So this is the overall process for cellular respiration. So the equation for cellular respiration is 6O2 plus C6H12O6 yields 6CO2 plus 6H2O and ATP. The purpose of cellular respiration is to harness the glucose and the oxygen and transform it into ATP, which is the universal energy currency in cells. And cellular respiration is important to the body because it's the process in which the body and plant cells create ATP, which powers pretty much all of the bodily functions and is essential to any cell's survival. The ATP is what provides the chemical energy that fuels all chemical reactions that happen inside the cell. So in this diagram, you can see the four main steps of cellular respiration. The blue square is glycolysis. The little bar between the blue and the red is the pre-Krebs cycle. The red-orange circle is the Krebs cycle, and the purple is the electron transport chain and ATP synthase action. The purpose of glycolysis is to break up a 6-carbon sugar glucose molecule into 2 3-carbon sugar pyruvate molecules to create ATP and to create NADH, which will help produce more ATP in the future. Glycolysis takes place in the cytoplasm of all cells. It's when a series of 10 reactions convert sugars such as glucose into pyruvate, NADH, and ATP. The pyruvate and the NADH created in glycolysis are used later to produce more ATP, and the NADH creates ATP through a process called oxidative phosphorylation. A total of 2 NADH, 2 pyruvate molecules, and 4 ATP molecules are made through glycolysis for every 1 glucose molecule. However, since glycolysis requires 2 ATPs for energy, the process has a net output of 2 ATP molecules. Glycolysis is also an anaerobic process, meaning that it occurs without oxygen. In the pre-Krebs cycle, you have the pyruvate acid becoming acetyl-CoA. To go from the pyruvic acid to the acetyl-CoA, CO2 is removed from the pyruvic acid and 2C acetate combined with the coenzyme A to produce acetyl-CoA. NADH plus gains a hydrogen and becomes NADH2, with a net result of 2 acetyl groups, 2 CO2 molecules, and 2 NADH2s. The Krebs cycle happens in the matrix of the mitochondria, and it's where 2 pyruvate molecules created in glycolysis become another 2 ATP molecules. For this to happen, one of the pyruvates is oxidized, which means that one of the carbon molecules in the 3-carbon pyruvate molecule bonds with oxygen, leaving behind a 2-carbon molecule, acetyl-CoA. The electrons in this stage, NAD plus and FAD, are enzymes that can hold on to electrons and hold the energy until the electrons need to be released. When the enzymes gain a hydrogen molecule, the NAD plus becomes NADH, and the FAD becomes FADH2. One pyruvate molecule produces 3 NADH molecules and 1 FADH2 molecule, yielding a total of 6 NADHs and 2 FADH2s per glucose molecule per cycle. And it's a cycle because the citric acid is oxidized, losing carbons over several steps, eventually returning to oxaloacetic acid. And 2 more NADH molecules are produced, which are used later to create more ATP. The Krebs cycle also produces CO2 as a waste product, which is exhaled by the organism. Additionally, the Krebs cycle is an aerobic process, meaning unlike glycolysis, it requires oxygen to happen. Citric acid is also a byproduct of the Krebs cycle, which is why the cycle is sometimes called the citric acid cycle. The electron transport chain happens in the inner membrane of the mitochondria, where with the inputs of NADH and FADH2, those molecules provide electrons which provide energy to power the electron transport chain across the inner membrane of that mitochondria. So the process starts with an NADH molecule entering the first protein in the chain from the matrix, oxidizing and splitting into NAD plus and 2 hydrogen ions, losing 2 electrons. Those 2 electrons then power the protein that pushes the hydrogen ion into the intermembrane space. The FADH2 enters the second protein in the chain of the matrix, splitting into FAD plus and 2 hydrogen protons, losing another 2 electrons. Those 2 electrons fuel the protein to push the hydrogen ions into the membrane space again. The increasing of hydrogen ions in the intermembrane space creates a concentration gradient that pushes the hydrogen protons from the outside back through the membrane through the ATP synthase into the matrix so that the balance between the outside and the inside of the membrane is equal. So with the concentration gradient pushing the hydrogen protons back through the inner membrane through the ATP synthase into the matrix to equalize the concentration gradient, it turns the turbine and causes the phosphorylation reaction of ADP plus P to create ATP. As the electrons released from the NADH and the FADH2 travel back through the ETC and pile up, they release into the matrix where O2 is, and then the oxygen in the matrix is reduced, making water, H2O, which is a product of the electron transport chain.